Imagine having a superpower that could pause time for a living thing, keeping it safe for years, decades, or even centuries. This might sound like something from a science fiction movie, but scientists are using a real-life version of this power to protect our planet's plants. This special technique is called cryopreservation. The word cryo comes from the Greek word for cold or frost, and preservation simply means to keep something safe. So, cryopreservation is the science of preserving living things, like plant seeds or tissues, by cooling them to extremely low temperatures. It is like putting them into a deep, deep sleep, from which they can later be awakened. The key to cryopreservation is using temperatures that are far colder than your freezer at home. Scientists use a substance called liquid nitrogen, which is incredibly cold, sitting at around minus 196 degrees Celsius, or minus 321 degrees Fahrenheit. At this frigid temperature, all the biological processes inside a plant's cells almost completely stop. Think of it like a movie that has been paused. Nothing moves, nothing changes, and nothing ages. This state of suspended animation allows the delicate genetic information inside the seeds or plant parts to remain intact and viable for a very long time, waiting for the right moment to be brought back to life. You might wonder why we can't just use a regular freezer for this important job. The problem with standard freezing is water. When water freezes slowly, it forms sharp, jagged ice crystals. These crystals can act like tiny knives, piercing and destroying the delicate cell walls and other important structures inside a plant. This damage, known as freezer burn on a larger scale, would kill the plant tissue, making it impossible to revive. Cryopreservation techniques are specially designed to avoid this crystal formation, ensuring the plant material remains unharmed and ready for future use, protecting its potential for regrowth. So, in simple terms, cryopreservation is not just about freezing, it is about freezing things very carefully and very, very quickly. Scientists use special cryoprotectants, which are like antifreeze for cells, to help protect the plant tissues as the temperature drops. This careful process ensures that when the time comes to thaw them, the cells can wake up without any damage. It is a high-tech method of creating a biological time capsule, a frozen library of life that holds the key to protecting plant species from disappearing forever from our world. Scientists who use cryopreservation are like modern-day Noahs, building a frozen ark to save plant species from extinction. Instead of herding animals 2 by 2, they collect seeds, shoot tips, or even tiny plant embryos. These samples are carefully prepared in a laboratory. First, they are often dried out slightly to reduce their water content, which is a crucial step in preventing those damaging ice crystals from forming. Then, they are treated with the special cryoprotectant solutions. Finally, these prepared samples are sealed in small, labeled vials and plunged into the icy vapor of liquid nitrogen for their long slumber. This frozen collection acts as a vital backup for our planet's biodiversity. Many plants around the world face threats from climate change, habitat loss, and disease. If a species goes extinct in the wild, its unique genetic code is lost forever. However, if we have its seeds or tissues safely stored in a cryobank, we have a second chance. Scientists can later thaw these samples, grow them in a lab, and potentially reintroduce the plant back into its natural habitat once conditions are safe. This makes cryopreservation an essential tool for conservationists fighting to protect the rich tapestry of life on Earth. Real-world examples show that this futuristic-sounding science is already making a difference. Scientists have successfully used cryopreservation to safeguard many rare and endangered species. For instance, several species of rare orchids, whose seeds are as tiny as dust, have been preserved using this method. Another success story involves the American chestnut tree, which was nearly wiped out by a devastating disease. Researchers have cryopreserved tissues from surviving trees, holding onto their resilient genes with the hope of one day restoring these magnificent forests. Each vial in the cryobank represents a story of survival. The process of bringing a plant back is just as meticulous as freezing it. When scientists decide it is time to wake a plant up, they must thaw it very quickly and carefully. This rapid warming prevents ice crystals from forming during the thawing process. The revived tissue is then placed in a special nutrient-rich gel in a petri dish. With the right conditions of light and temperature, the tiny plant part can begin to grow, first into a small seedling and eventually into a full-sized plant. This process, called tissue culture, allows a single preserved sample to give rise to a new generation, While cryopreservation offers incredible hope, it is not without its challenges. The biggest enemy, as mentioned, is ice. Even with the best techniques, the risk of ice crystal formation is always present, especially in plant tissues that naturally contain a lot of water. Some plants, particularly those from tropical regions, have large, water-rich seeds that are extremely difficult to dry out without killing them. For these recalcitrant seeds, standard cryopreservation methods do not work well. Scientists are constantly experimenting with new cryoprotectant formulas and flash freezing techniques to overcome this fundamental hurdle and expand the range of species they can save. Another significant challenge is the high cost and technical expertise required. Building and maintaining a cryobank is an expensive endeavor. The large insulated containers that hold the liquid nitrogen, known as DWARs, require regular refills. The laboratories need specialized equipment, and the scientists and technicians must have extensive training to handle the delicate plant materials and hazardous liquid nitrogen safely. Because of these costs, cryopreservation facilities are not as widespread as they need to be, particularly in developing countries that are often home to the highest levels of biodiversity and the greatest number of threatened species. Furthermore, not all plants respond to cryopreservation in the same way. Each species is unique, and a technique that works perfectly for one plant might fail completely for another. This means that researchers must develop a specific, tailor-made protocol for almost every new species they want to preserve. This process involves a great deal of trial and error, taking up valuable time and resources. For thousands of endangered plants, the specific methods for successful cryopreservation have not yet been figured out, creating a race against time as their populations in the wild continue to decline. Finally, there is the challenge of long-term viability. While we believe that samples stored in liquid nitrogen can remain viable for hundreds or even thousands of years, this science is still relatively young. We do not have definitive proof that extends over such vast timescales. Scientists must conduct regular tests, thawing a small number of samples every few years to check if they can still grow. This ongoing monitoring is essential to ensure that our frozen arks are truly secure and that the precious life stored within them will be ready when we need it most in the future. Saving plant life is about much more than just protecting pretty flowers or majestic trees. It is about protecting ourselves and the health of our planet. Plants form the very foundation of most ecosystems on Earth. They produce the oxygen we breathe, provide food for humans and animals, and create habitats for countless creatures. When a plant species disappears, it can set off a chain reaction, affecting all the other organisms that depended on it. By using cryopreservation to prevent extinction, we are helping to maintain the delicate balance of nature that sustains all life, including our own. This frozen library of plant genetics also holds immense potential for the future of agriculture. As our climate changes, farmers face new challenges like drought, new diseases, and extreme heat. The wild relatives of our crop plants, many of which are rare or endangered, often possess valuable genes for resistance to these very threats. By preserving them in cryobanks, we are saving a treasure trove of genetic traits. Scientists can study these preserved plants to find genes for drought tolerance or disease resistance, and then use that knowledge to breed hardier, more resilient crops that can feed a growing global population. Furthermore, plants are a vital source of medicines. Many of the drugs we rely on today, from aspirin to cancer treatments, were originally derived from plant compounds. It is estimated that tens of thousands of plant species have yet to be studied for their potential medicinal properties. Every time a plant species goes extinct, we might be losing the cure for a future disease. Cryopreservation ensures that this incredible botanical pharmacy remains available for future generations of scientists to explore, potentially leading to groundbreaking new treatments and therapies that could save countless human lives. In the end, cryopreservation is a powerful symbol of our responsibility as stewards of the Earth. It is an act of foresight and a promise to future generations that we will do everything in our power to pass on a world as rich and diverse as the one we inherited. These frozen gardens are not just collections of dormant seeds, they are banks of hope. They represent a safety net for biodiversity, a resource for human innovation, and a testament to our commitment to preserving the beauty and complexity of the natural world for all time. Protecting plant life is not just an option, it is essential for a healthy and prosperous future.